All right, welcome everybody. So this webinar, as you all know, it will focus on the new version or newer release of OrthoMCL 7, and we'll cover some of the cool features and functionalities that this resource now has. Um, Before we get started, a couple of technical things. So we are using just a Zoom meeting, so feel free to, hopefully everybody's familiar with Zoom by now, but feel free to, if you have questions, feel free to raise your hand during the webinar. You can use the raise hand functionality if you want. If you can't find it and you feel like you must ask the question right then, feel free to unmute yourself, and we're happy. We welcome interruptions. We'd rather answer the question as we go along than wait until the end, or maybe you'll miss the opportunity to ask the question. Also, feel free to type the question in the chat, and we will be monitoring the chat and answering questions as we go along. So my name is Omar Harb. I'm the senior project manager for the ViewPath project. I'm joined by a few members of the ViewPath team, including Stuart Brown, who actually run the majority of this webinar and walk you through OrthoMCL 7. I'm also joined by Nipur Katur and Evelina Bashenko, who will be monitoring the chat and helping answering some questions and making sure we are addressing any issues that come along. Before we get into OrthoMCL 7, I wanted to walk you through a couple of slides. So OrthoMCL is part of a family of databases that are run and managed and developed by the ViewPath project, which many of you, I think, are familiar with. These include many databases that cover various parasitic organisms, fungal pathogens, and model organisms, vectors of disease, including things like mosquitoes and ticks, databases like HostDB that cover host response to pathogen infection, clinical and microbiome databases that are associated resources that are built on the same model. And finally, OrthoMCL, which is a comparative genomics resource, which will be the subject of today's webinar. And we heavily rely on OrthoMCL for integration into ViewPath and all of our databases. And so it's highly relevant, not only if you're interested in basic comparative genomics across the tree of life, but also if you're specifically interested in a particular parasite or a fungal pathogen or a vector, and you're interested in finding genes that are a particular phyletic pattern or looking for homologs or orthologs across various organisms. Before we get into the details of the webinar, many of you are probably aware that ViewPath recently moved towards a subscription model. The subscription model is to encourage various groups to get a subscription to keep the resources open access. Our goal is to keep the resource free and available to everyone, but I'm sure everybody can appreciate that these resources cost money to keep going, and we need to raise the money somehow. And so one aspect, we'll continue writing grants and getting funds from other sources, but this must be supplemented. And in fact, we're hoping that it'll be completely sustainable by user subscriptions. We have a lot of information on resources. I won't focus on this. We have additional webinars as well that cover the subscription model, and we have a lot of material that will help you navigate the subscription model, including a comparison between what a typical subscription would cost and other things that we typically are used to paying for in a lab setting. And so this should not be viewed as any differently than that. Back to our webinar, and before I turn it over to Stuart, I wanted to highlight a couple of places where we utilize OrthoMCL within ViewPath resources, and then we will transition to talking about OrthoMCL and some really cool features that have been developed recently and made available in OrthoMCL 7. So I'm looking at ViewPath the parent site. All of our databases are built on the same infrastructure, which means that anything you learn in one of the sites, you can apply to the others, which is quite nice. You can search for genes based on their specific phyletic pattern. And I'll demonstrate this quickly. We have other webinars that focus on this particular tool, and also some YouTube shorts, which allow you to look at this. But right here on the left, I'm going to look for the search for a phyletic pattern. So I'm just filtering my searches. and I looked for a search that allows me to find genes based on their phylogenetic profile. When I select this, I can very quickly go in here and ask for which organisms I'm interested in returning. In this case, I'm selecting Apicomplexa. So these include things like Plasmodium or Toxoplasma. And I'm more interested in things that are conserved in Apicomplexa. So I'm going to click once on this and select a check mark. And I'm interested in things that are specifically also conserved in Chlorophyta. So one of the plants, basically. These include many other organisms underlying these, right? So I'm just clicking the entire family here. Once you click on Get Answer, this will give you a return result. So this returns 708 genes that are conserved across all Apicomplexa and within plants. And so you get this list of genes, and now you have your phylogenetic profile or genes that meet that phylogenetic profile. The other place where we integrate OrthoMCL results is within gene pages. So I'm just going to go ahead and go to one of these gene pages. This is in Babesia, the Gemini, I believe. And so I'm on this gene. It's a DNA-directed RNA polymerase. I can scroll down to this gene page. Again, gene pages are very detailed and full of a lot of information. But we have a section on orthology and synty. And if you go to this section, you will find a table here that shows you all the orthologs within PyroPlasma-DB in this case. So all the Babesia orthologs or paralogs and homologs. And scrolling down, you will see here there's a section called synteny, which allows you to look at the collinearity of genes across all the organisms within PyroPlasma-DB. Actually, this only shows a sampling of them. You can tailor this to show the synteny across different organisms, but the shading here, that relies on OrthoMCL. So this is shading the orthologs along this collinear region of the genomes. And so this is a place where OrthoMCL gets integrated. I mentioned this table up here. And then finally, we have links that allow you to go and visit the group that this gene belongs to in OrthoMCL. And since OrthoMCL7 was just released, we keep OrthoMCL6 available. There was a big change in the algorithm, which I'm sure Stuart will let us know about that. But as a result, we're keeping the old version around because there are some differences between group structures between OrthoMCL6 and OrthoMCL7. Okay, with that, I'm going to stop sharing and turn it over to Stuart and let you take over with the rest of the presentation.
All right, thank you, Omar. I'm going to share my screen and try and do this whole thing live the way you've been doing rather than just having slides. Looks good. So a few general points. First, I guess I should introduce myself. Stuart Brown. I'm the team leader for the group that's been developing this new version of OrthoMCL. I'm a bioinformatician and I work at UPenn for the ViewPathDB project. Our goal in producing a new version of OrthoMCL was to update the data because it had gotten old, improve our algorithm so that we could get the compute done more efficiently. It had become very difficult for us to recompute the ortholog groups because of the size of our database. We have millions of proteins, and doing all against all comparisons with BLAST was just too slow. And it was taking too much compute power, and we were producing new versions too slowly. We developed a new algorithm and a new clustering method based on OrthoFinder, which is a well-regarded method for building ortholog groups that has done well in the Quest for Orthologs competition. And one of the key things in OrthoFinder is that the comparisons are done with Diamond, which is an accelerated alternative, drop-in alternative to BLAST. So similarities computed with Diamond are still considered to be BLAST similarities. It's just with an accelerated method. It still produces z-values that are comparable. There's a slight loss of sensitivity at the extreme end, but much less than 1%. So you lose the very least similar things, which is not usually the case for an ortho group anyway. One of the advantages of OrthoFinder is that it uses phylogenetic inference in building its groups. And we have updated the look of our ortho groups to include a phylotree of all the proteins in each group instead of the older cluster diagram. And we've also added some new tools that make it easy to find orthologs between species and to map a file of proteins to the OrthoMCL groups. Another thing that we did with the new version is we updated the genomes. The older version had a lot of duplicated genomes because it was difficult to recompute. When a reference genome was updated in the public domain, we weren't able to remove the old proteins and add new ones because it would change the cluster. So we duplicated the genome, and we had about 50 of those. And so it made the clusters kind of inaccurate. There were duplicates of identical genes coming from the same species just based on a different build and annotation of the genome. So we eliminated all the old ones, and now we only have the most recent version of each reference genome. Now OrthoMCL downloads from UniProt the most recent version of each genome every time we do a build, which we anticipate will be at least every 6 months, perhaps more often than that. We now have 149 core organisms, which form the basis of our orthogroup clusters. We had to eliminate Trichomonas because of its super high number of internally duplicated genes, which were taking far too long to compute with our current methods. So that's now what we call a peripheral genome. It's not one of the genomes that we use to build the core clusters. It's one of the genomes that we use to add proteins to the clusters. All right, I mentioned that we use Diamond. We use OrthoFinder. OrthoFinder normalizes the e-values that are computed, the blast values, both for the length of the proteins and for their phylogenetic distance. The consequence of this is that the groups are going to be different between OrthoMCL6 and OrthoMCL7, our current version. The new version is also slightly more cohesive. The groups are somewhat larger, more inclusive than in OrthoMCL6, but not 100% of the time. Sometimes the algorithm will adjust the e-values and it'll end up splitting some proteins from an old group into a new one, which is one of the reasons why we make it pretty easy to check back and forth between the old groups and the new to see where your proteins of interest have been located. And as far as I've been able to tell, it's always logical. There's always a gray zone where you could split or you could bunch these proteins together. And given the density of the cluster, it makes sense to split them where they were. All right, so this is the new OrthoMCL homepage. It looks very much the same as it always has. We've got these cards here that give you short bits of information. We have the help that takes you to some longer tutorials. And we have the search tools on the left here for ortholog groups and proteins. The searches are also up here in this menu. We have reduced the number of tools. It's a little bit cleaner. I'll get to those in a minute. And actually, the first thing I want to do is similar to what Omar was doing, is that I want to show you how to get to OrthoMCL from ViewPathDB. So this is the, oops, well, that was a bummer. Hopefully I'm still sharing. Now I'm sharing the ViewPath screen, and you can type in the name of your favorite gene, whatever that might be. This is an Anopheles gene. So when you ask for this gene, it's going to give you a link to this gene, and it's going to take us to Vectorbase. And then if you scroll down a little bit on the side here, there's the orthology section. And at the top of the orthology section is the link to the old group, the legacy group in OrthoMCL6. And this will take you to this protein and its group in OrthoMCL7. We have this little navigation menu on the side, but I usually click it closed so that I've got more screen to show the main features. And I want to do a little tour of the OrthoMCL group page. So at the very top, we have this sort of summary section. We tell you how many proteins are in the group, how many are part of that core of 149 species that we use to seed the clusters. Then the rest are peripherals, meaning additional organisms where we add the proteins to those core clusters. Keywords is super important. That tells you basically a summary of the description, those 420 proteins, the words that you find most often in the protein descriptions. It's sort of an AI thing. It's not perfect, but if there's if it's a cytochrome C oxidase COX-20, then many of those proteins are going to have that name, and it's going to show up here in keywords. The top PFAM domains. So it'll only show PFAM domains that are present in several of the proteins. If there's just like one protein that has one extra PFAM domain tacked on the end, that's probably not going to show up here. And then previous groups is basically links back to the Ortho6. This next section we call phyletic description. It's a taxonomic overview for this group. What are the organisms that this group is present in? So in this case, it's in fungi and metazoa. There's a couple in amoeba and a few in this sort of catch-all group that we call other eukaryotes, and they seem to come from the Mycota, Pythium and Phytophthora and those sort of things. There's actually a hidden feature here called hide zero counts. If you uncheck this thing, you get to see where this group is not present. So there's none of these in plants, none in archaea, and none in bacteria. And none in alveolates. So if you're looking for Plasmodium, it's not there. This gene is not present there. So that's actually kind of a cool feature, and you can use that in reverse when we do what we call a phyletic search, meaning you can search for groups that are present in certain taxonomic categories and not present in others. So, you could look for, say, genes in fungi that are not present in mammals as a, possible target for some type of chemical or that sort of thing. Another feature that's new for OrthoMCL7 is what we call similar groups. This is a cytochrome, COX-20. There's 420 proteins in this group. There's another group that has 10 proteins that's also COX-20. So this is a case where the algorithm split off a group for whatever reason, and you can see the reason right here. The E value, the blast E value between these two groups is only 0.02. Actually, it's more, or yeah, it's less significant than 0.02. So that means that it doesn't meet our stringency for clustering, but in fact, these two groups are related. And in terms of the PFAM domain, it's the same, right? They've got 12597, and this one is also 12597. So that's inevitable that when you set up a somewhat arbitrary boundary for what makes a cluster, there's always going to be some things that fall outside. And similar group is a nice way of tracking that and saying, if we relax the stringency a little bit, what else might fall into this ortho group? EC numbers, well, there's no EC numbers because those are enzymes, and this is not an enzyme. PFAM domain, 100% of our proteins, well, no, I think there's 420, 404 of them have this PF12579 cytochrome C PFAM domain. All right, now we come to the most visible feature of our new Ortho 7, which is this phylogenetic tree merged with a protein table. In this case, we have 420 proteins in the table. It's a bit much to be able to scan this tree and understand it. So we've added this set of filters on the side that I believe make it a lot easier to work with the tree. The first one that I usually test out is limit only to core organisms. So in this case, you know, if there's more than one species in a genus or several organisms that are closely related, only one of them is probably going to be in the core. And so this drops it down to 62 rows. And a few things become obvious when we limit it a little bit this way. And one of the interesting ones is that a lot of these genes are paralogs, meaning they've been recently duplicated within a particular lineage. So this is chicken over here, and it's got a bunch of genes, and they're all very closely similar to each other and not super closely related to anything else. It's like it's in the same general branch as the mammals, but not super close. Then there's situations where, well, there's one gene from a dog that's really close to this one in whales and cows. And then there's another here. So this is a gene family that's duplicated, you know, further back in the mammalian lineage. And so we have this idea that you can, you know, check off the proteins that are interested to you. And if you want, you can just filter down the tree and look only at proteins that you've chosen. So you can modify the tree, you know, in a lot of ways. Another feature that we've built in here is that if you select a group of proteins, you can run Clustal Omega on that group of proteins. So it'll just send this straight to a Clustal job. All right, I'm going to do one more tree. I mean, one more. And so there's our Clustal job for that group. And you can see that they're very cohesive for this domain, but this one gene is much longer and has a section here that's um, not matching up with the other ones. All right, so I'm going to go to one more group that I think illustrates some of the properties of ortho quite well. And oops. I use the site search a lot to get to groups. It does take 2 steps, but it's pretty quick. So this is a nuclear receptor subfamily 2. It's a really big group, 900 proteins. If you look at the phyletic description, it's only in metazoa, right? We've got arthropods, chordata, nematodes, but no plants, no fungi, no small eukaryotes. It's strictly in metazoa. It becomes really interesting to look at the similar groups. This is a nuclear receptor subfamily 2. And if you look over here, we have a nuclear receptor subfamily 4, estrogen receptor, nuclear receptor group A, uh, retinoic acid receptor. So there's clearly a superfamily of nuclear receptors that all share some pretty significant amount of blast similarity to each other, all at the sort of e to the minus 20 level. And so everything that's grouped inside this particular ortho group is more similar to each other than that. Also, if you look at the PFAM domains, many of these similar groups share PFAM group 104. But if we had grouped all of these things into just one ortho group, we'd have many thousands of proteins and they would form subgroups within that one group. And you'd say, well, maybe we need to split those out into their subgroups. So the algorithm makes sensible decisions, although not everyone agrees with them all the time. There's this summary of the PFAM domains, which is interesting. There's 900 something proteins in this group. 700 of them have the zinc finger domain, and almost 700 also have this ligand binding domain. And then these other domains are only tacked on to just one protein, so they're not particularly relevant for the group. And then if you look at the table, almost every protein has both of these PFAM domains in it. And they tend to be in the same structural organization. There are a few proteins where one of those domains is missing. Those are shorter proteins. So there's some evolutionary process where these shorter ones got chopped off, and they are, in fact, you know, they've been grouped on the tree, so they're close to each other phylogenetically. Again, 900 is too many to really understand what's going on here in terms of protein evolution. So one of the things I could do is trim it down to just the core. That's 255. It's still a lot. Maybe too many for me to really start building a story here, but I actually know something about this nuclear receptor subfamily. It's been published on quite a lot, and I know that Drosophila has some proteins in this family that have, you know, a lot of experimental data behind them. There's one called tailless, there's one called dissatisfaction, and there's one called 7-Up. And yeah, and one of the things I thought was interesting, and I just thought I'd follow up on for a second here, let's add the human and say, how do human proteins relate to Drosophila proteins? And now we can see that tailless is a close ortholog with the human nuclear receptor subfamily true 2 groupie member 1. We don't have a catchy name for the human gene. At least I don't know one, but maybe there is one. HGNC. But these two are actually, they form a subgroup. It's pretty obviously cohesive. We could say, is it the same in mouse? We could add mouse here and say, does mouse also have that exact same protein as the tailless one? And you could say, yeah, here's tailless, here's human, and here's mouse. They form a subtree. And interestingly, Drosophila has 2 more in that subtree that are not really, there's no paralogs in human or mouse. So we don't know when that duplication happened, but seemingly Drosophila has its own thing going there. All right, now I wanted to look at the 7-Up isoforms. That seems to be only in Drosophila. There's no other proteins in this little subgroup of 7-Up. So I said, well, let's see what goes on in another arthropod. We have a lot of anopheles in our database. Let's add a couple of anopheles and see what happens then. Did that work? Yeah. And they didn't show up. Huh. Oh, I have to turn the core only off. There they are. Okay, so those are the Anopheles versions of 7-Up. And they're sort of in the same group. Well, yeah, 7-Up. And so Anopheles has some copies that are form the same group with the Drosophila 7-Ups. A lot of them haven't been named, so this particular organism doesn't have as in-depth gene annotation as the Drosophila, which is one of the uses for OrthoMCL, is to transfer annotation between model organisms and those that have been less well studied. All right. Did I get through everything? Yeah, I wanted to keep going a little bit down the
Stuart, can you, before you go further, can you, somebody is asking about if you have, if the group is more than 1,000 members in size, what happens to the tree? Is there a way for them to get the tree, or what options do they have?
well, what you have to do is trim it down to less than 1,000. We've just said that that tree is too big to handle. And the way I most typically do it is I click on core only, and that'll trim it down. If your organism is not part of the core, then you'll have to work with the filters to find some subset of the proteins that is relevant to you, maybe add 6 of your favorite species and get a tree that has less than 1,000 proteins in order to draw the tree. The one other option is that the trees exist even if we're not drawing them. And at the bottom of the table, the bottom of the list of proteins, we have a button that says download the raw Newick file, and that will give you the entire tree no matter how large it is. And then you can take that Newick file over to any visualization software you want, like ITOL, or just any tool that can draw a Newick file, and then you can play with the tree on your own. So thanks for the question. I should have pointed that out. Yeah, actually, that was the exact moment that I wanted to stop for questions because this ends up being the bottom of our
Good.
ortholog.
Yeah, we've been answering a number of them online and chatting the responses. Um, I think we are probably good to go on, unless anybody who's been answering questions or anybody who's asked questions needs further clarification, or if you haven't asked your question and would like to ask it by unmuting yourself, that's fine as well. There's another question maybe right now before we move on. So Mike Mathers is asking if there's a statistical measure of the support for the branches computed in the um, in OrthoMCL. So like Bootstrap or anything like that in the tree or
Not
in
bootstrapping the trees because some of our groups are extremely large, like more than 10,000 proteins, and bootstrapping that would add sort of days and weeks to our compute. And we're not, we made the choice not to show bootstrap values because we didn't want to do that much computing. For the really small trees, obviously we could have done it, but
and
it's
is the improved statistics, Stuart, group statistics, would that provide some help in this scenario?
not really. The group statistic is what it's doing is it's showing you the dispersion of the group, meaning
Hmm.
how how tightly clustered they are. And it's really only interesting and meaningful when you can compare different groups. So like if you have a group that's more tightly clustered versus one that's kind of dispersed, that's sort of a catch-all that's throwing things in. So I agree that, phylogenetic trees ought to have bootstraps, but it was beyond the ability of us to do that for 250,000 groups.
Yep. Hmm. Great. Thanks, Stuart.
All right, I was just going to show a few of the search functions. Uh, Here on the main OrthoMCL page, we have group and protein searches. Um, you can search in the groups or the proteins for a text term, synthetase, if that's what you're interested in. We have the wildcard asterisk. Um, like the rest of UPathDB, any sort of search that's created with the search function is automatically the start of a search strategy, which means that you can then combine text with any of the other tools that we have. So let's say you look for synthetases and you wanted groups that had at least um, at least 5 or at least 10 proteins in them, but no more than 1,000. And so of those 4,000 groups that we found, now we have 2,000 that have, between between 10 and 1,000 members. So the idea is you could also add a phyletic constraint here. So you could say, oh, I'm only interested in groups that, contain some metazoa, but no fungi, and add that step. All right. And there you go. So of those 2,000, now I'm down to 253. Other searches that might be interesting are searching by a PFAM domain. Um, because ortho is not one-to-one with PFAM, most of the time PFAM domains are larger than a single OrthoMCL cluster. Orthologs, there can be multiple genes that have the same PFAM domain, and true orthologs are somewhat tighter than that. Some PFAM domains are extremely general. They're in, a huge number of proteins. but you can find all of the ortho groups that contain your PFAM domain of interest. And yeah, and then you also have this constraint here, which sort of adds another search at the same time. You could leave out, for example, all the singletons, which I often like to do. If something falls in an ortho group of one, that may be some kind of annotation error, or it could be extremely interesting, unique protein that is only in one organism. So that you have the option to filter the groups at that point without having to do a second step of the strategy. But that particular PFAM domain is in 31 ortho groups. So it's a pretty widespread PFAM domain, and some of these groups are probably not similar groups to each other, meaning they don't share enough similarity to show up in our similar groups algorithm. And I think we've shown phyletic pattern a couple times, so I'm going to skip over that. And I want to show two brand new features for OrthoMCL. One of them in the tool menu is called Map Proteins to OrthoMCL with Diamond BLAST-P. We used to have a Galaxy tool. We don't run a Galaxy anymore, which did essentially this same thing. You have a file of proteins. Generally, they would be the whole proteome from some newly sequenced organism. Could also be an assembly of transcripts, RNA-seq, run an RNA-seq, do an assembler, find coding sequences and translate them. There's a tool that can do all of those things. But then you have the heavy lifting of annotating many thousands of putative proteins. And this is basically going to do it in one click. You just choose a file that has, say, some translated coding sequences, either from a genome or transcriptome, metagenome, load it in here, give it a name, and hit map. Now this thing takes, well, it usually takes 5 to 10, maybe even 15 minutes to run, but I did this same job yesterday. It recognized that I was running the same query sequence, and it took me to one of my saved jobs. Every time you run a blast job, if you're logged in to OrthoMCL as a registered user, which I am, it keeps a running backlog of all of your previous blast jobs. And so when you run the blast job on the screen, we show your top 100 hits. Actually, not your top 100 hits, your first 100 hits. So the first 100 proteins in your query file are shown here with their e-values. And we have specifically chosen to keep those proteins in your data file that have no match. So we throw a group ID of -1 here and a matching query of an asterisk. So that query sequence has no significant match anywhere in OrthoMCL. And given how comprehensive OrthoMCL is, it's questionable whether that's a real protein or not. I mean, it might be, but you would probably want to flag that up and say, maybe this is not good. If your query file has a lot of those, you might say, how did I build this file of proteins? Is it full of errors? So that's helpful. In addition, we show a particular protein that you could look up in ViewPath, which is the best match, what group it is, a summary that is actually drawn from the protein ID. And so it's not quite as good as the group summaries that we have for OrthoMCL, because a lot of proteins in genomes don't have great annotation. Some do, some don't. So it might be most helpful to save this column and look up each one of these ortho groups if you wanted to annotate your protein to the, with the most accuracy. Only the top 100, but what we do is we provide this whole file as a download. So you get a tab delimited file for every one of your query sequences, the best OrthoMCL match and what group it's in, and this description, which is drawn from the protein. Just one more thing I want to show, another brand new feature, which I thought might be useful to some people. Shared ortholog by organism. So if you've got, say, a genomics assay on some particular organism, which is not very nicely annotated, and you don't have great geo annotation on the organism where the genomics experiment was done, and you want to translate that over to better annotated reference genome, and a model organism that has good geo annotation. Instead of just doing a blast and taking the best match, we let you use OrthoMCL to say, find me the orthologs between each gene in this organism and each gene in this organism. And very quickly, it'll give you an answer that there are 1,961 proteins in your query organism that have a group that have an ortholog in your target organism. Now, there could be a many-to-many situation where several genes in one organism match the same, are in the same ortho group. If I sort by ortho group, that'll be more obvious, right? A whole bunch of genes in this organism are in the same ortho group, and they match the same set of genes in the other organism. So to summarize that, what do we have to do here? We have to add a step and transform into groups and run that step. Now we've condensed the 1,961 proteins into 1,256 proteins, each one in a unique ortho group. So this is the ortho group mapping for your queries to your target. All right, so that's all I had to say. I'm going to reopen it for questions from anybody. You can unmute and speak up. And maybe I'll keep the screen share. What the heck, in case somebody wants something demonstrated.
Stuart, I see a question in the chat about the Diamond Blast output. Does 0.0 for the e-value indicate a super small e-value?
Yeah, it does. That's Diamond's default. For things that are smaller than e to the minus 200, it defaults to 0. I think Blast does the same thing.
Stuart, another question maybe you can demo is, what does the add column option do? And so as you're doing that, we have this add column option on all of our sites. You can view this table of results as a dynamic spreadsheet, basically, that you can add any data we have in the resource as a column.
In this case, add column doesn't give you a heck of a lot, because by default, we've turned on almost everything.
Mm-hmm.
I don't know where to go, but I actually left behind something pretty important here. You can scroll to the right and see a lot more info that we have about each ortho group. Oops.
And you can also drag and drop columns and move them around.
You can reorganize the columns. Exactly. but yeah, if
David, go ahead.
Yeah, Stuart, I just wanted to raise a couple of other questions that have come up in the chat. And for those who are not following the chat, there are many questions there along with answers I'd encourage you to take a look at. So there are a couple of additional questions. There's a question from Michael Mather asking whether there's a statistical measure of support for these branches comparable to bootstrap values. And I wonder if you want to comment on MCL.
I already answered that. We are not saving, storing, computing the bootstrap values because of the size of the compute. We're doing 250,000 groups, and to bootstrap them would escalate the compute by many fold.
And there was also a question about the construction of phylogenetic trees, which Omar has posted an answer to for how trees are generated. Do you want to comment on how these trees compare to the trees that are generated by the OrthoFinder algorithm?
We use exactly the same method as OrthoFinder. We're doing multiple alignment with MAFT, and we're doing the trees. I'm spacing out what we do with the trees, but they are approximately. Oh, sorry. I'm not going to come up with the right words right this second. I'm gonna. Let me look up where I've written it down.
We have some more information in the FAQ in our frequently asked questions section under help, and so there is, for that question and many other ones, we have some information. Also, there's an analysis method section where we have even more detailed information. But yeah.
And just a reminder to all participants and any users of ViewPathDB resources, you can always click on the Contact Us link on any site that enters into a tracking system. So your response will get to the right person, whether that is Stuart or someone else, and we will get a response back to you by email.
For those of you interested, we have some slides for this presentation, but most of this was done as a live demo. But we have recorded this webinar, and probably later today we will send an email out with a link to the recording. We'll make that link available to everybody who attended, anybody who registered, but also to anybody else who would like to watch it on our YouTube channel. So,
got my answer. These are called approximate maximum likelihood trees that are computed with FastTree.
Mm-hmm.
And this is,
and I see Alejandro also said the same thing in the chat. So yeah, you're getting some.
And this is the same method that's used by OrthoFinder, although we're actually computing it ourselves, not relying on OrthoFinder to compute the trees. Again, because of the size of the compute, we can manage it better on our cluster, doing it ourselves. And actually,
great, I think.
Yeah.
Go ahead. No, continue.
And the trees that you see on the screen are drawn with the tidy tree R library that we have actually extended custom ViewPath code, and then the ability to redraw them in real time is something that we've created custom for this website with the filters.
Somebody did note that they were trying to download the Newark file for a really big tree over 14 big orthology group over 14,000 proteins, and there was a note on the page indicating that there was some we hit an issue, so there was a bug. It turns out that only affects. I did some testing. It only affects orthology groups that are bigger than 14,000 members, so they're not that many.
But there's maybe two that are that big.
Yeah, there may be a little bit more, but it's affecting those only. So we'll look into that and see what
we will
the
definitely
issue is.
look into that. I mean, yeah, we could recompute those couple of trees and maybe zip the Newark files so they don't hit a browser bug.
Right. Okay, we're approaching the end of the hour. Are there any other questions? People feel free to raise your hands or type them in. And as usual with all of ViewPathDB resources, We love to hear your feedback. So as you use OrthoMCL 7, if you come across any issues, if you notice a group that looks funny, something is weird about it, we'd love to hear about that. Sometimes there are really good logical explanations for why a group looks a certain way, and sometimes there aren't, and it's good for us to see those. Also, if you have additional feature suggestions, we love to hear those suggestions, as those help us develop and tailor the sites to things that our users need and can use. And so, in fact, a lot of the things that you see on ViewPath2B resources are built based on user feedback and input, and that helps us develop things that make sense and are useful to you. There's a question from Peter Olila. Can domain shuffling or rearrangement, maybe due to evolutionary mislead phylogenetic reconstruction?
Absolutely. It's impossible to perfectly resolve the shuffling of domains with any single set of rules. So what we're doing is based on sequence similarity over more than half of the length of the proteins. And in some cases, there's going to be merging. In some cases, there's going to be splitting that an expert on that particular protein family might not agree with. You know, we do approximately the best we can, and the algorithm that we use, you know, can make errors, or at least it can make, what would you call it? You know, it makes a call as best it can.
Right.
Let me also suggest two other processes. So Stuart's answer on that is certainly accurate, but two strategies that you might want to pursue if you are interested in domain reorganization, for example, in metabolic cluster genes in fungi, or in other proteins that are reorganized in various ways. One is, as there was some discussion of this in the chat earlier, you might want to use to explore methods that focus more on domains rather than entire proteins, such as eggnog. And the second is that you might want to explore genes, for example, in your species of interest in OrthoMCL, by searching for PFAM domains and comparing those yourself, which might very well highlight cases of genes which have clearly been rearranged, for example, in polyketide synthases or other such proteins.
Great. Well, it doesn't look like there are any more questions, and we're at the top of the hour. So thank you, everyone, for webinar. As I mentioned, this is recorded shortly. Let us know if you have any questions, as usual, and see you soon on a future webinar. Take care.